It's now time for Cape Biology. I am Joanna George Johnson, and in today's lesson, we are going to take a look at photosynthesis. So, here begins. Today, we are going to be looking at photosynthesis, and we now have the definition. We are going to be looking at to define photosynthesis. We're then going to be outlining a particular section or stage of photosynthesis called the light-dependent stage. And then we hope that by the end of this lesson, you'll be able to appreciate that the purpose of the light-dependent stage of photosynthesis to the light-independent stage of photosynthesis, which happens to be the second stage of photosynthesis. So here we begin. Just to let you know, most persons would have been familiar with photosynthesis already because they would have done photosynthesis in grade 10. However, the topic of photosynthesis in grade 13 is a little bit more advanced. Usually we know already that photosynthesis is about the process of autotrophic organisms, mainly plants, as we know, to have the ability to make food using basic substances known as carbon dioxide and water. There is oxygen that is also made. There's oxygen that is also made during the process. And typically, from back in CSEC days, this is the equation that you would use where you have 6 carbon dioxide molecules adding to 6 molecules of water, and in the presence of sunlight and chlorophyll produces glucose and oxygen gas. However, this equation could not be far from the truth of really what happens with photosynthesis in a typical plant. Because actually what happens is there's no time during the process where carbon dioxide and water ever meets. Okay? So what happens is that photosynthesis is broken up into 2 different stages. It's called the light-dependent stage and the light-independent stage. Now, as the name suggests, one stage requires light. Clearly, it will be the light-dependent stage because it depends on light. And the other stage does not require light. Okay? It is the light-independent stage. Today, we are going to be focusing on the light-dependent stage of photosynthesis. So, when we are looking at anything in biology, it's a trick that I use all the time. The trick is, what is happening? Where is it happening? How is it happening? And also, why is it happening? So, today we are going to be more focused on how is it happening and why it's happening and where. Okay? So what is it? What is photosynthesis? Or what is the light-dependent stage of photosynthesis? The light-dependent stage of photosynthesis is a stage of photosynthesis that requires light. Okay, as the name suggests. Where does it happen? Well, we are going to take a closer look. Everybody knows photosynthesis occurs in the chloroplast. But today we are going to take an even closer look at the chloroplast and find out that within the chloroplast you have a particular structure known as the thylakoid membranes, and that is where the light-dependent stage of photosynthesis takes place. Then you have the situation, you have when does it happen? Clearly, if it's the light-dependent stage of photosynthesis, it has to happen when there is light around. Bright light preferably. That's the reason during the day when there's bright sunshine, maybe not like today, but generally in Jamaica we have bright sunshine, you have a lot of photosynthesis going on because of the bright light that is out. Why do we need, or why is the light-dependent stage of photosynthesis important? The light-dependent stage of photosynthesis is important because it is the stage that makes ATP and NADP and pH, and is used, both of these substances are used in the next stage of photosynthesis, which is the light-independent stage. And we will look closer at these later on. How does it happen? We are going to be focused on that for most of today's lesson, because this is the stage where students tend to have issues with photosynthesis at the CAPE level. And we're going to be looking at three things: light harvesting, photophosphorylation, I know it's a real word, trust me, and also we'll be looking at chemoosmosis if time permits. So now we are going to be looking back at our board. We are still on the topic of photosynthesis and on the topic of the light-dependent stage of photosynthesis. Let me emphasize. So now we are going to focus on the where. When we are doing photosynthesis originally, everybody knows, you ask a question to any student from even grade 7, where does photosynthesis take place? In the green leaves. But then if we go further, as we advance through biology, we realize it's not just in the green leaves, but certain sections of the green leaves, because there are some cells in the green leaves that have chloroplasts, which is the actual site of photosynthesis in leaves. However, at this advanced stage of biology, we take an even closer look at the chloroplast, where we actually look inside of the chloroplast and what it looks like inside. Now, chiefly inside the chloroplast is a whole connectivity of membranes, okay? These membranes are called thylakoid, okay? And thylakoid membranes contain the pigments that are necessary for photosynthesis. That's where we find them. Now, outside of the thylakoid membrane, still within the chloroplast, and by the way, this is the diagram that you normally would see in an exam, so that's one of the reasons I placed it here, because you need to be familiar with the type of diagrams expected in the exams. Now, around the thylakoid membrane, in the space in between the thylakoid membranes, you have what's called the stroma. The stroma is like a gel-like substance, mainly made of water, contains of enzymes, proteins, and so on, that also contribute to the whole process of photosynthesis. However, for today's class, we are going to be focusing on the thylakoid membranes, because that is exactly where the light-dependent stage of photosynthesis takes place. All right? So let us move on to why. Why do we need the light-dependent stage? Well, the light-dependent stage of photosynthesis is where these two main substances, ATP and NADPH, are made. Now, ATP and NADPH are very important to the next stage of photosynthesis, hence why the first stage is important. And the bonus of this stage is that oxygen is also liberated. And a little tip, oxygen, all the oxygen in the world that exists today, that is very important for all living organisms, actually are produced from the process of the light-dependent stage of photosynthesis. Okay, so we move on very quickly to the how. Now, this is where we're going to be spending most of the time, because this section of photosynthesis happens to incorporate a lot of chemistry. And I do know that there are some students who are having an issue with chemistry from time to time, and so because of that, we are going to spend the time to ensure that the topic or this stage is fully understood. Now, if I was to ask, what are the pigments found in chloroplasts? What are the pigments that are found in chloroplast? I could hear right now persons sounding off, yes, chlorophyll, miss, chlorophyll. Chlorophyll. But guess what? That is just the tip of the iceberg. Actually, in the chloroplast, you have several other pigments available. You have xanthophylls, you have carotenoids, and even so, you have more than one type of chlorophyll. You have chlorophyll A and you have chlorophyll B. And chlorophyll A, actually, there are different types of chlorophyll A, which we are going to be getting up close and personal with today. Okay, and this is very important. Now, the pigments, all of those pigments I just mentioned, are are found in the thylakoid membrane of the chloroplast. Now, these pigments are not just placed in the thylakoid membrane just haphazardly, just throw down. No. They are actually in a particular formation called a photosystem. They are arranged in a particular arrangement, and so that are called the light harvesting complex. Now, what exactly it is a large protein molecule, and you should be familiar with the fact that proteins are always embedded into membranes in cells, and this is one such protein, a protein complex, and within the protein complex, you have all of these pigments, the xanthophylls I mentioned earlier, carotenoids, chlorophyll A, chlorophyll B, and the like. And within the reaction center, the light harvesting complex or the photosystem, you have where light from the sun or from some other light source may hit the pigments. And when they hit the pigment absorb this light at different wavelengths or at different levels. Now, all of these pigments absorbing at different levels, they get very energized. And when they get energized and get excited and they're moving around and vibrating, guess what? They trigger neighboring pigments beside of them to get energized as well. And they continuously do this in a haphazard way until it reaches what's called the reaction center. Until it reaches the reaction center. Now, at the reaction center is a specialized chlorophyll A molecule. It's always chlorophyll A. Different photosystems have different chlorophyll A molecules embedded at the reaction center. All right, we will talk more about that shortly. But what happens is when all of this energy from all the neighboring pigment molecules go to the reaction center, reaches the reaction center gets so excited that 2 electrons from that particular chloroplast gets removed or jumps out from the actual chlorophyll molecule. Now, what happens is when that excitement or excited electrons is removed from the chlorophyll molecule at the reaction center, they have one of 2 options. One, because everything that goes up must come down, right? But how they come down makes the difference. See, normally in a regular, if you had some chlorophyll in a beaker or a test tube, what would happen is that the electrons will just come back down, and because they are going to be attracted back to the reaction center. And when the electrons, as they come back, make their way back down to the reaction center, what would happen is they will just emit energy. As they lose the energy, because they're coming back to a stable area, while as they lose that energy will be emitted as light and heat. But in the confines of the thylakoid membrane, in a photosystem, the electrons that escape the reaction center is actually captured by what's called a primary electron acceptor. Now, the primary electron acceptor okay, is a particular molecule, and at this level you do not need to know the name of the molecule, so we are going to leave it at that. Now let us look at this whole happening in context of the light-dependent stage of photosynthesis, okay? Now here we have what is commonly called the Z scheme, okay? The Z scheme comprises two photosystems, photosystems 1 and photosystems 2. Yes, they did not bother to give any fancy names to them. And just to let you know, the reason why one is called photosystems 1 and the other is called photosystems 2 is because photosystem 1 was discovered first and photosystem 2 was discovered afterwards. So that's the reason for the nomenclature. Now, as I said earlier, we will now be looking at the whole process of the light-dependent stage as it occurs within the photosystems on the thylakoid membrane. Remember, all of these molecules are embedded in the thylakoid membrane of the chloroplast. And clearly, being a chloroplast, it will have several of these whole systems throughout the entire chloroplast. So let's start with photosystems 2. Even though, you know, it's called 2, as I said before, 1 was just found before 2. But everything really starts at 2. As I said before, the accessory pigments at the photosystem, they all receive, you know, light, absorb light coming in from the sun or whatever the light source is, and eventually they pass all this energy to the center, the reaction center, which is, as I said before, the reaction center is a particular chlorophyll A molecule. Now in photosystem 2, that particular chlorophyll A molecule has a name. The name is P680. They're called P680 because that is the wavelength of light that this particular chlorophyll absorbs light at. Okay, 680 nanometers. But they call it P680 for short, and we will do so too. Now, when P680 gets excited because of all of this energy coming from the accessory pigments, which are the other pigment molecules in the system, and also from the sun, whatever energy it gets from the sun, what happens is that 2 electrons get promoted to a higher energy level. That promotion of the 2 electrons to the higher energy level is accepted by the primary electron acceptor. Now, the primary electron acceptor holds those electrons so that they don't just come back to the reaction center, because then that would not have offered any help for photosynthesis. Instead, what happens is that as the primary electron acceptor accepts these 2 electrons, it means now that P680 is short 2 electrons, and it wants it back. But it cannot now get it from the primary electron acceptor because the primary electron acceptor is holding on tightly to these 2 electrons. So instead, P680 needs to seek to get these 2 electrons from somewhere else. And where is it getting these electrons from? It's going to get it from water. Now, when you're going back to grade 10 or grade 11, when you did photosynthesis first, you'd have heard the term photolysis. Now, photolysis means literally the splitting of water by light. And commonly held by most students, you know, at that level is that the water is split by light during photosynthesis. Technically, that is not really what happens. What really happens is that P680 so wants back its 2 electron that is just lost, that it actually has that strong pull of electrons towards itself from the water molecule. But because in the position that the water is in at the time, with all of this light and all around, its such a stable molecule becomes weakened, the bonds of it becomes weakened because of the pull of the P680 that really wants its 2 electrons back. So what happens is water is split into hydrogen ions, and oxygen gas will eventually be made when enough water, because obviously this is happening millions and millions of times in each chloroplast, in each cell, in each of the leaves in a plant. So you know it's a lot of reactions going on. But apart from just the 2 hydrogen ions that are released and the oxygen that comes off, there are 2 electrons that also comes off during the splitting of the water. And so therefore these 2 electrons go to satisfy P680. To satisfy P680, so P680 feels good again, it has its 2 electrons, it's all right. But then you see those 2 electrons that the primary electron acceptor accepted already? Guess what? It is going to pass it on to a next electron acceptor, which is going to pass it on to a next electron acceptor, and so on and so on and so forth. As this happens, this chain of electrons being transported from one carrier to a next carrier to a next carrier, the electron that is in this very excited state still loses energy. Now you have to recognize that when the electron is in this high energy state, some electrons, some compounds or some molecules can't hold them. So that's the reason specific carriers, which are actual molecules, will be carrying these 2 electrons and then pass it on to a next one, then to a next one, and then to a next one in this chain. While this is happening, all of this energy that the electrons are losing as they go from molecule to molecule to molecule, ATP, the form of energy, is made. The ATP is synthesized through this process. Now that process has to be explained later, but we are going to move on to now photosystems 1. Now, while all of this excitement going on at photosystem 2, photosystem 1 actually has its own excitement going on. The same thing, light is hitting the different molecules in the system, and the molecules are getting all excited, and they're moving up, and they're hitting into each other, transferring the energy to other neighboring molecules, and eventually hitting the chlorophyll A at the reaction center. Now, the chlorophyll A at this reaction center of photosystems 1 is called P700. So I want you to notice, P680 is the reaction center for photosystems 2. P700 is the reaction center for photosystem 1. And this is a crucial mistake students make from time to time. So please do not make that mistake. Please, I beg you. But anyway, same thing happens. P700 gets so excited, 2 electrons go up, and, well, they don't come back down because the primary electron acceptor accepts it, accepts them, and does a similar situation of passing the electrons from one molecule to a next molecule, all the time losing a little bit of energy. Now, when this P700 loses those 2 electrons to a higher energy level, what happens is that the P700 now wants back its electron just like P680. But this time, P700 don't have water to help it out. Instead, what happens, those 2 electrons that came from P680 has now been passed on from molecule to molecule to molecule and are now heading to P700 because they are now attracted to P700 because P700 has lost its 2 electrons to the primary electron acceptor and now requires the 2 electrons. So the 2 electrons coming from P680 eventually ends up at P700. All right? And then, after this is satisfied, what happens here is that there's no photosystem 3. for collecting the electron that came from P700. So instead, this is what happens. If you notice that this chain of electron transport is a little bit shorter than the first one that we looked at. And that is because the molecule NADP, and by the way, it is an official acronym, we can use it. It has a name, a longer name called the nicotine adenine diphosphate, but we are not going to use that. You just remember NADP and you're good. If you notice, NADP is the final electron acceptor of the P700 electrons that left it. And when it accepts it, it comes back here. See those two hydrogen ions that came off when water was split? It joins with the NADP to form NADPH. Now this, remember I said earlier that there are two main things that are produced from the light-dependent stage of photosynthesis, and they were ATP and NADPH. Now I want you to notice something. Most of us are familiar with ATP. We know that ATP is the energy currency of living organisms, of living cells. But a lot of us may not be familiar with NADPH. I want you to take a note that the electrons came from this level, from P700, was shot up to a higher energy level. But notice that the NADPH that is formed from collecting those two electrons are still not at the same level as where P700 is. It means here that NADPH right now has a high energy, is of high energy, and therefore has the ability to be an energy source in the next stage of photosynthesis. And so here we have it, starting from P, for photosystems 2, with P680 getting all excited and losing 2 electrons. And then those 2 electrons get passed on from molecule to molecule to molecule until it reaches P700, where photosystem 1 is. And P700 already had lost 2 of its electrons because of getting excited from the light and being accepted by a next primary electron acceptor, and then passed down to several molecules till eventually making NADPH. But please note that NADPH has collected these 2 electrons at a higher energy level compared to where these electrons started out, which means NADPH has a very high energy bound into it. Therefore, it, along with ATP, can provide the energy needed to fuel the process of the light-independent stage, which is the next stage of photosynthesis. Now, I'm going to try to explain chemoosmosis to you, because that is how ATP is made. Okay? And I'm going to do this very quickly. Ooh. Okay, we are here. Now, believe it or not, this is the exact same thing that I just explained, but this time it is showing the arrangement of the various molecules and systems in the membrane itself. Please remember that in the chloroplast, you had the stroma, which was just the gel-like substance, and then you had the lumen, which is the cavity in between the 2 membranes in the thylakoid membrane. So what happens is when water gets split and these 2 hydrogen ions, the hydrogen ions are given off, eventually these hydrogen ions are going to accumulate in the lumen of the thylakoid membrane. And as you know from Unit 1, things that accumulate on one side of a membrane wants to go to the other side by diffusion. Now, because this is hydrogen ion and it's a charged particle, you know it can't just pass through the membrane just so, because it will be repelled by the phospholipid bilayer. So instead, there is a particular protein called ATP synthase that these hydrogen ions must go through in order to go to the other side. So when they go through, as they go through this particular protein, they make ATP on the way out and at the same time be able to add to NADP and the electrons to make reduced NADP. Okay, so we are going to stop here for today. Sorry I didn't get to go through in more with you, but I hope that you have been enlightened by how we have gone through this topic. So that is all the time we have for K-Biology today.